have a richer understanding of the dynamics, not only the B factors, but also how those motions are correlated. Okay, so this is not a new idea. So this field of diffuse scattering from proteins is probably about 40 years old. And so actually a lot is known about diffuse scattering from proteins. On the other hand, you can show all the studies that have been done on one slide. So, so the very early work was really very revolutionary. In particular, this one, everyone should read this paper by Kaspar. In many ways, we're just following up on this early result. When I started doing this, which was actually around 2016 is when I started, I had all these questions like, what is causing diffuse scattering of proteins? How should data be processed? What is the contribution of our internal motions that we care about versus other kinds of disorder? What models should I use to interpret the data? So actually, the field had not converged on answers to any of these questions. It was still very unknown. Okay, so what we reasoned is that we needed to just collect the best possible data we could get, try and figure out answers to some of these questions. And in particular, we realized that a lot of the controversy came about because it was not clear the role of lattice disorder versus internal motion. And to really separate those two things, you need to measure very large length scales. And that means sampling the map of reciprocal space very finely. So our goal was to collect these high quality, detailed reciprocal space maps. Okay, but it was a good time to start that because we had pixel array detectors available, and they're really ideal for diffuse scattering. So they have this narrow point spread function, high dynamic range. You can do fine fly slicing, which is all of you know about now. These are very standard. This was just a crystal of lysozyme. You can see, Bragg peak is like a few pixels wide, and we get 5 orders of magnitude dynamic range, just moving a few pixels away from the Bragg peak. So this is great. And then we also chose a model system that was well suited to this question. That was lysozyme in the triclinic space group. So the reason we chose P1, there's no symmetry averaging of the diffuse pattern, so it's easy to interpret. This crystal had very low mosaicity, high resolution diffraction, small unit cell. That means the Bragg peaks are far apart. So you get lots of samples. And we could grow pretty large crystals, which is important because we wanted to measure the room temperature. So the data collection that we did, there was nothing particularly fancy about it. It was, I would say, very low dose, high multiplicity, fine phi slicing, all the best practices for room temperature. And it was necessary in our case, to merge from 3 crystals, 11 wedges of 50 degrees. And the only thing we did that was really different is we collected the background scattering for every phi angle of the spindle. And that's because the shadow of the pin kind of wanders around on the detector as you rotate. And then so we can do then a frame by frame background subtraction. Okay. And then there was a lot of work on software, and one of the things we had to kind of reinvent was scaling. So basically, we did an aimless style scaling model refinement. But there's a few extra things we had to include, and one was a bit of added background scattering. So because the protein is on a loop, as you rotate, at some point during the rotation, the loop and some extra solvent will move through the beam, and so you get a bit of added isotropic background. So this scaling model kind of takes care of that. And at the end, we merge in 3D, and we have